introduce our next speaker now, and let me share my little slides here. Could you see the slide that I have?
Yes, we can.
Okay, so our next speaker is Dr. Pedro Jose from George Washington University, and I think I have to do this. Okay. And there's his picture. Still a very handsome man for a 78 years old gentleman. So Dr. Jose got his MD degree from the University of Santo Tomas. I did not get when, but some time ago. And he has the honor of placing first in the Philippine National Board examinations in medicine and surgery. And then he got his PhD degree in physiology at Georgetown University. And one of the distinctions is that he defended his dissertation with high distinction. Dr. Jose's current position is as professor of medicine in pharmacology and physiology at the George Washington University. That's a university close by in the White House. He has been a visiting professor in several universities in China. He has guest lectured in many universities all around the world. And he was also the PAASE 2005 co-awardee in science, just like Dan today is a co-awardee for that. He's the past president of the American Society of Pediatric Nephrology, published a lot of papers, at least 380 papers, and has a very high H index of 57. Now in 2018, Dr. Jose got one of the highest awards that the president of the Philippines gave, and this is the Pamana ng Pilipino Presidential Award for Filipino individuals in organizations overseas. And there is the President of the Republic putting the ribbon and the medal on Dr. Jose. And this is for exemplifying the talent in industry of the Filipino. They brought the country honor in this recognition through excellence and distinction in the pursuit of their work or profession. So this is a very high honor for Dr. Jose. We're very proud of him. And it's my pleasure to introduce him to give his talk, which will be on gastro-renal communication, its role in hypertension. Pedro, so let me unshare here. Second, yes. Okay, go ahead. Pedro, you're muted. Unmute yourself.
Mute.
Yeah, click on that red microphone to unmute.
There.
Okay, thank you very much. Go
Can
ahead.
you hear? Sure, the slides are there?
The slides are here, yes. Thank you.
Okay, that's what I was worried about. Okay.
Okay.
Thank you. And I thank your invitation. I'm going to be talking about gastro-renal communication role in hypertension. In order for zero sodium balance to occur, the amount of sodium that is ingested must equal the amount of sodium that's excreted, mainly in the urine. When the excess sodium that is ingested is not excreted by the kidney, and the excess sodium retained in the body is not buffered in the interstitial space and lymph, blood pressure increases. We nephrologists believe that the kidney is the most important organ of the body, especially in the regulation of blood pressure. And that is according to Homer Smith, the father of modern nephrology, who wrote a book called From Fish to Philosopher. And he said that our kidneys enabled us to evolve. In the beginning, the ocean was isotonic, nutrition diffused in and waste diffused out and diffused back in. As the salinity of the oceans increased, a distal tubule formed to eliminate sodium and waste products of metabolism. Driven by wanderlust, we migrated to fresh water. The environment changed from hypertonic to hypotonic state. We needed to get rid of the water. Therefore, we developed glomeruli to eliminate water, but sodium from the filtered water had to be reabsorbed, so proximal tubules appeared. When we migrated to land, the problem changed from overhydration to dehydration. Therefore, we developed a thick skin and decreased our glomerular filtration rate to save water. However, the decreased filtration rate decreased our ability to excrete waste products, and so we decreased our metabolism. But the low metabolism impaired our ability to react quickly. One who is slow cannot rule the earth. We increased our metabolism, but the waste products had to be eliminated. Therefore, high glomerular filtration rate returned. To prevent excessive excretion of salt and water, we developed the loop of Henle. The apes became humans, and we ruled and continue to rule the earth. Homer Smith said, bones can break, muscles can atrophy, glands can loaf. Even the brain can go to sleep without immediate danger to survival. But should the kidneys fail, neither bones, muscles, glands, nor brain or life itself could carry on. Oops, that was before dialysis and transplantation. So what organ regulates blood pressure? Sherlock Holmes said, too many suspects. Narrow them down, Mr. Watson. The problem is in the kidney. How does one prove that the kidney regulates blood pressure? Shown here is the kidney, and a functional unit of the kidney is called a nephron. We have about 1 million nephrons per kidney. There are 3 ways: surgical transplantation, combined surgical and molecular biological manipulation, molecular biological methods, for example, renal selective modification of gene of interest. First, we talk about surgical transplantation. Blood pressure in 6 African Americans with malignant hypertension was normalized after renal transplantation, and this was reported 37 years ago. Hypertension developed in the recipient of a kidney from a hypertensive donor, but without a family history of hypertension. Transplantation of kidneys from normotensive rats to nephrectomized spontaneously hypertensive rats, or SHR, prevents the increase in blood pressure, while the converse increases blood pressure. And cross-renal transplantation in dull salt-sensitive rats to dull salt-resistant rats and vice versa causes salt-sensitive or salt-resistant hypertension. The blood pressure is not, in hypertensive strains, is not normalized with transplanted kidneys from normotensive strains. This indicates the importance of extra renal mechanisms in the regulation of blood pressure. The next method is combined surgical and molecular biological manipulation. How can one determine the importance of the kidney in the regulation of blood pressure? And one is by studying the effect on blood pressure of cross-renal transplantation in a knockout mice with hypertension and their wild-type counterparts. Transplanting one knockout kidney to a wild-type body with no kidneys, and transplanting one wild-type kidney into a knockout body with no kidneys. This is the recipient mouse. We get rid of both kidneys of the recipient mouse, and then we transplant one kidney to the recipient mouse. And these studies were done by Dr. Lara Asico in the laboratory. And there will be 4 groups. You see in here, donor kidney transplanted to, that is wild type, that is transplanted to a wild type recipient body. A wild type donor kidney transplanted to a knockout recipient body. A knockout donor kidney transplanted to a wild-type recipient body, and knockout donor kidney transplanted to a knockout body. We reported that deletion of the dopamine 5 receptor gene increases systolic blood pressure in mice. Here's the systolic blood pressure, and these are the wild-type mice, and the blood pressure of the D5 receptor knockout mice minus is elevated, and these are unmanipulated mice. Now we transplant wild type to wild type and knockout to knockout, and you can see in here that the blood pressures of the wild type to wild type transplants are the same as those of the wild types that are unmanipulated, and so are the mice with the knockout to knockout have blood pressures that are elevated, similar to the unmanipulated dopamine 5 receptor knockout mice. Now, transplantation of a knockout kidney into a wild type body increases systolic blood pressure, and transplantation of a wild type kidney into a knockout body decreases systolic blood pressure. Renal cross-transplantation experiments indicate that blood pressure regulation is 80% renal and 20% extrarenal. The third method is molecular biological method, that is renal selective modification of gene of interest. So here, you can manipulate the gene of interest in the renal cortex by the renal subcapsular infusion of siRNA or silencing RNA. Or you can also do retrograde ureteral infusion of agents to manipulate the gene of interest in the renal tubules only. Silence or knock in the gene of interest in nephron segments. You can see here that adeno-associated virus-mediated renal gene transfer by retrograde infusion via the ureter. These are in vivo bioluminescence images. We retrogradely infused only into the left kidney, and so here in the ventral position and dorsal position. Here, the adeno-associated viral-mediated renal gene transfer by retrograde infusion via the ureter and immunostaining for the detection of the fluorescent GFP in the mouse kidney. And you can see here that we can have the distal tubule, and you can hear that we can see the proximal tubule of the kidney. Ingested sodium by the stomach or gut is one mechanism by which sodium balance is regulated. After ingesting too much salt, the stomach begs the kidney, please rectify my dietary indiscretion, and we call this the gastro-renal axis. Several gut hormones have been proposed to mediate the natriuresis or increase in sodium excretion following an oral sodium load, and these enterokines or GI hormones or gut hormones. The examples are uroguanilin in here, cholecystokinin or CCK, and gastrin. And these hormones increase sodium excretion. Gastrin is secreted by G cells in the antrum of the stomach, duodenum, and pancreas. It stimulates secretion of gastric acid by the parietal cells of the stomach and aids in gastric motility. Circulating gastrin levels are 10 to 20-fold higher than cholecystokinin or CCK levels. Circulating CCK, proguanilin, pro-uroguanilin, and uroguanilin levels are not increased following ingestion of sodium chloride. By contrast, an increase in sodium intake increases circulating gastrin levels. Of all the gut hormones, gastrin is the one that's taken up to the greatest extent by renal proximal tubules. By secreting gut hormones like gastrin in response to ingested sodium, the stomach directs the kidney to increase sodium excretion. A certain amount of sodium in food increases serum gastrin levels. This is 30 minutes post-ingestion in anesthetized, salt-resistant BAVC mice. You can see here serum gastrin level, basal state. Very little sodium, nothing happens. But when one increases the amount of sodium in the food, you can see in here that serum gastrin levels increase. Now, sodium independent of food can increase serum gastrin levels, again in BAVC mice. Here, distilled water only, a little bit of sodium only, and a little bit more of sodium. And you can see here the increase in serum gastrin levels. The presence of food, however, increases the ability of sodium to increase serum gastrin levels in biopsy mice. You can see in here, sodium only, but when one adds food, you can see the marked increase in serum gastrin levels, and a little bit more if the sodium concentration is increased in the stomach with or without food. How can we be sure that the sodium-mediated increase in serum gastrin level is not caused by neural mechanisms? So what we did is to determine the effect of sodium and food introduced to the stomach ex vivo. See, here is the ex vivo stomach, in which the esophagus in here is ligated with a tube going into the stomach, and the pylorus is ligated, as you can see here. The expression of gastrins increased in mouse stomach exposed to sodium chloride. Now, sodium is mainly responsible for the increase in gastrin expression in mouse stomach exposed to sodium chloride ex vivo. You can see here sodium chloride. We get rid of the chloride, but sodium is still there. And you can see here that sodium gluconate increases gastrin expression just as efficiently as sodium chloride. When one gets rid of both sodium and chloride and use mannitol instead, again, same osmolarity, almost nothing appears. And then when one gets rid of sodium and chloride still there, gastrin expression is also not increased. Now, gastrin and annexin 4 in human G cells in here, shown in here. This is gastrin in red, is annexin in 4. And the reason I'm showing you this is because annexin is important in the exocytosis of proteins. This is the plasma membrane. Here you have gastrin and annexin. You have the yellow in here, red and green. And then when one merges all of this, gastrin, annexin, and plasma membrane, you can see in here the white, that's the gastrin undergoing exocytosis. We next studied the effect of blood pressure in the germline deletion of gastrin in salt-resistant biopsy mice. Here's the expression of gastrin in wild-type mice, ++, see there, and very little, if any, in the gastrin knockout mice. You can see in here that the blood pressure of gastrin wild-type mice is not affected by a decrease in sodium intake or an increase in sodium intake. However, in the gastrin knockout mice, these gastrin knockout mice have high blood pressure, and the blood pressure is decreased by a low salt diet and increased again by a high salt diet. Notice in here that the normal sodium blood pressure is actually the same as that in the high sodium diet, so that if one only does an experiment in which one tests only the effect of normal sodium and high sodium in blood pressure, then the erroneous conclusion would be that that mouse or animal or human, if you can do that, is not salt sensitive. What causes the increase in blood pressure in gastrin knockout mice? Well, you can see here that the natriuretic response, this fractional sodium excretion here to an oral sodium load, is impaired in gastrin knockout mice, the open bars. And there are several sodium transporters in the kidney, and the renal sodium hydrogen exchanger type 3 or NHE3, sodium potassium 2 chloride cotransporter NKCC2, and epithelial sodium channel ENaC in here, and sodium potassium ATPase expressions are increased in the gastrin knockout mice. However, the interpretation of the effects of germline deletion of the gene of interest is confounded by compensatory mechanisms during development unless the gene deletion is inducible. To determine the role of gastrin released by the stomach and duodenum and sodium excretion after a meal with sodium, we devised a method to silence gastrin secretion by infusing gastrin siRNA into the celiac artery. This method also provides direct proof that the stomach communicates with the kidney or any organ of interest. Here is the mouse stomach in here. Here is the aorta. Here's the celiac artery, and we put in a catheter in here, celiac artery, and kind of thread it close to the gastroduodenal artery. Here's the expression of gastrin in the mucosa of biopsy mouse infused with MAC siRNA. Gastrin still there, but almost completely gone with the silencing siRNA. Now, as it turns out, gastrin mRNA or gastrin is also expressed in other organs beside the stomach. You can see in here, renal medulla has a little bit, renal cortex has a lot, and adrenal medulla, and of course the stomach should have a lot of gastrin. So we then infused the gastrin siRNA into gastroduodenal artery, and it decreases, see here, gastrin mRNA, but not in the kidney, which means that the infusion is selective to the stomach. Systolic blood pressure under anesthesia is increased in CCKBR mice. Cholecystokinin receptor is the receptor of gastrin. You can see in here the increased systolic blood pressure. Systolic blood pressure under anesthesia is also increased in these mice, wild-type mice, treated with a CCKBR antagonist. And you can see in here that systolic blood pressure is also increased in the gastrin wild-type mouse treated with the gastrin siRNA infused into the gastroduodenal artery. And the natriuretic response, or FENA, to an oral sodium load is impaired in gastrin knockout mice and the gastrin siRNA treated gastrin wild-type mice. There is a problem. Gastrin is important in
we lost you, Pedro.
He might have trouble with his internet connection.
Yeah, it looks like he's frozen. Richmond?
Yes, sir.
What do we do here?
Do we
put
the presentation to the other presenter, po, sir? Or, and we'll just go back to Sir Parlo when he's okay with his internet.
Let's wait 1 minute. He might be reconnecting.
He did say there was a storm passing by, so I'm wondering.
Yeah, so maybe it's in the Virginia area now, you know. So, although I think it was passing by in South Carolina at this moment in Charleston.
Can somebody call Pedro? Leah? Dan, do you have his number, cell phone number? You may not be aware that he cannot be heard.
Yeah, he disconnected.
So maybe somebody can call him or text him. But I think many of us know that there are storms in the East Coast of the United States. Yes.
Yeah, so the other possibility is we could have Carlito present his talk. And then if Pedro comes back, then he could finish it after Carlito.
Okay, we'll wait for another 2 minutes and then
yeah.
Carlito. Yes.
Yeah, it's very possible that this storm is messing up his connection somehow. So Carlito